Space travel accelerates aging in human stem cells, reveals study. A groundbreaking study has revealed that space travel can significantly impact human stem cells, causing them to age at an accelerated rate. Researchers found that spending time in microgravity on the International Space Station alters the behavior of blood-forming stem cells, leading to signs of premature aging. This discovery has significant implications for long-duration space missions and the health of astronauts. The study, conducted by scientists at the University of California, San Diego, involved exposing human bone marrow cells to the unique conditions of space for 32 to 45 days. Upon their return, the cells exhibited classic signs of accelerated aging, including increased energy consumption, loss of rest state, and the activation of normally silent DNA segments. These findings were published in the journal Cell Stem Cell. The research team, led by Catriona Jamieson, MD, PhD, focused on hematopoietic stem and progenitor cells, which are crucial for maintaining a healthy immune system. They used palm-sized bioreactors to grow these cells in orbit and tracked their behavior with onboard imaging. The results showed that stem cells in space lost their normal functions, struggling to renew or regenerate, which is a critical factor for astronauts' long-term health. One of the most intriguing findings was the surge in activity from repetitive DNA sequences, known as their dark genome. These elements, usually quiet, became active under the stress of microgravity, indicating unusually strong cellular stress. Scientists also observed changes in inflammatory signals and mitochondria, which are part of the cellular stress response that can lead to aging. Microgravity, the weak gravity experienced in orbit, disrupts the way cells sense force and organize themselves. Combined with space radiation, this environment appears to accelerate the cycling of blood stem cells, depleting metabolic reserves and disrupting normal repair processes. Over time, this pattern mimics the aging process that occurs over many years on Earth. The study also uncovered clues from telomeres, DNA caps that protect chromosomes and shorten with age. The team detected a decline in telomere length and changes in the expression of genes responsible for maintaining telomeres, suggesting stress on genome maintenance. Some cells even carried mutations associated with clonal hematopoiesis, a condition linked to increased risks of cardiovascular disease and leukemia. Interestingly, when these space-exposed cells were cultured on a young, healthy support layer, some of the damage was reversed. Self-renewal improved, and stress-related gene activity returned to a calmer state. This finding suggests the possibility of designing countermeasures to restore rest and repair pathways, potentially helping astronauts before and during their missions. The study's findings align with previous research, including a landmark year-long astronaut study that showed shifts in gene activity, immune responses, and telomere dynamics during time in orbit. Many changes persisted after landing, highlighting the long-term effects of space travel on the human body. The Space Omics and Medical Atlas, an open resource, integrates multi-omic astronaut data, revealing common threads of mitochondrial stress, inflammation, and DNA stability. These findings emphasize the importance of monitoring stem cell health during long-duration missions to prevent potential health risks for astronauts. Furthermore, the study's insights could lead to the development of therapies for patients on Earth whose stem cells show similar stress. The research team plans to sample cells during future space missions to better understand the effects of radiation, microgravity, and sleep cycles, providing valuable knowledge for both space exploration and terrestrial medicine.